Chapter 5: Antigen Recognition by T lymphocytes. The T-cell receptor resembles a membrane-associated Fab fragment of immunoglobulin. This figure compares the structures of an antibody and a T-cell receptor, showing both shared features and key differences. At the top, the antibody is shown with two identical antigen-binding arms and a constant stem region. Each arm includes a variable region formed by one light and one heavy chain. Below, the T-cell receptor consists of two chains, an alpha and a beta chain, each with variable and constant regions. This structure resembles a single arm of the antibody. Unlike antibodies, which can be secreted, the T-cell receptor is always membrane-bound. It includes a transmembrane segment and a cytoplasmic tail for signaling. The T-cell receptor does not bind free-floating antigens, but recognizes processed peptide fragments presented by major histocompatibility complex molecules. Structurally and functionally, the T-cell receptor is similar to a membrane-anchored fragment antigen-binding region of an antibody. This three-dimensional structure shows a T-cell receptor made up of two chains, alpha and beta. Each chain contains a variable region at the top and a constant region at the bottom. Together, the variable regions form the antigen binding site. The overall shape and domain arrangement are similar to one arm of an antibody. The beta chain is shown in blue and the alpha chain in purple. Loops in the variable regions, labeled 1, 2, and 3, are the complementarity determining regions. These loops interact directly with the antigen. This structural similarity reflects the shared evolutionary origin of T-cell receptors and antibodies, even though they recognize antigens in different contexts. T-cell receptor diversity is generated by gene rearrangement. This figure explains how the diversity of T-cell receptors is created through gene rearrangement, a process similar to that used by antibodies. At the top, the alpha chain gene is located on chromosome 14. In its germline form, it contains multiple variable and joining gene segments. Through recombination, one variable and one joining segment are selected and joined, forming a functional gene. After transcription, splicing, and translation, the final alpha chain protein is produced. At the bottom, we see the beta chain gene, located on chromosome 7. It undergoes an additional recombination step that includes diversity gene segments, resulting in a variable diversity joining combination. Once both chains are produced, they come together to form the complete T-cell receptor. The variable regions of each chain combine to create the antigen binding site. This process allows T-cells to generate an immense variety of receptors, each with a unique specificity for recognizing antigen fragments presented by other cells. This slide shows the clinical consequences of defective T-cell receptor gene rearrangement. On the left is a child with severe oral candidiasis, a fungal infection that occurs when the immune system is unable to control microbial growth. On the right is a newborn with a widespread rash and signs of systemic infection. This presentation is characteristic of severe combined immunodeficiency. In such cases, failure to produce functional T-cell receptors results in a profound immunodeficiency, leaving the body defenseless against common pathogens. These images highlight the essential role of T-cell receptor diversity in protecting against infection and supporting immune system function. The RAG genes were key elements in the origin of adaptive immunity. This figure illustrates how the recombination activating genes, also known as RAG genes, help give rise to the adaptive immune system. On the left, a transposase enzyme from a transposable element inserts into a gene that encodes an innate immune receptor. In the center, the inserted transposon splits the receptor gene into two parts, flanked by repetitive deoxyribonucleic acid sequences. Next, chromosomal rearrangements move the transposase genes, now known as RAG1 and RAG2, to a separate location in the genome. These genes became essential for somatic recombination, enabling the assembly of diverse antigen receptor genes in lymphocytes. The table on the right shows that the recombination activating genes now function in several loci, including T-cell receptors and immunoglobulin genes across 5 different chromosomes. This evolutionary event was a turning point in the development of adaptive immunity, allowing organisms to recognize and respond to an unlimited range of pathogens. Expression of the T-cell receptor on the cell surface requires association with additional proteins. This figure illustrates how the T-cell receptor is expressed on the surface of a T cell and how it functions in coordination with other proteins. The T-cell receptor is composed of an alpha chain and a beta chain, which together form the antigen recognition site. However, the T-cell receptor alone cannot transmit signals. It requires association with the CD3 protein complex and the zeta chains for full function. The CD3 complex includes epsilon, delta, and gamma subunits, which are responsible for stabilizing the receptor at the cell surface. The zeta chains, shown in green, extend into the cytoplasm and are critical for initiating intracellular signaling once the receptor binds antigen. Together, these associated proteins allow the T-cell receptor to recognize antigen and send signals into the cell to initiate an immune response. A distinct population of T cells expresses a second class of T-cell receptor with gamma and delta chains. This figure compares the two major types of T-cell receptors found in the immune system. On the left is the more common alpha-beta T-cell receptor. It consists of an alpha chain and a beta chain, each with a variable region, a constant region, a transmembrane domain, and a cytoplasmic tail. On the right is the gamma-delta T-cell receptor, found on a distinct population of T cells. It follows a similar structural layout but contains gamma and delta chains instead. Both types of receptors form antigen binding sites using their variable regions. However, they recognize different types of antigens and are found in different tissues. Alpha-beta T cells are the dominant form in circulation and lymphoid tissues, while gamma-delta T cells are more common in epithelial and mucosal barriers. Together, these two receptor types contribute to the flexibility and breadth of the adaptive immune response. This figure shows the genomic organization of the John loci for the gamma and delta chains of the T-cell receptor. On the left, the delta chain locus is embedded within the alpha chain locus on chromosome 14. This region contains variable, diversity, joining, and constant segments. When a T cell commits to becoming an alpha-beta cell, recombination of the alpha chain deletes the delta chain locus, preventing both receptor types from forming in the same cell. On the right is the gamma chain locus, located on chromosome 7. It contains multiple variable and joining segments, followed by 2 constant regions. Both gamma and delta chains undergo gene rearrangement during T-cell development, similar to the alpha and beta chains. This arrangement highlights the shared evolutionary origin of the T-cell receptor families and reinforces the importance of gene recombination in creating receptor diversity. This table compares the genetic components that generate diversity in immunoglobulins and alpha-beta T-cell receptors. Immunoglobulin-heavy chains use around 40 variable segments and 23 diversity segments. In contrast, the beta chain of the T-cell receptor uses about 52 variable segments and only 2 diversity segments. Light chains and alpha chains do not use diversity segments. However, the beta chain frequently reads its diversity segments in 3 reading frames, adding even more variation. The number of joining segments varies. The alpha chain uses 61, while the beta chain uses 13. Immunoglobulin light chains use 5 or 4, depending on the type. The use of additional nucleotides at the junctions between segments, called N and P nucleotides, also contributes to diversity. This process is common in both immunoglobulins and T-cell receptors. Overall, the number of possible variable gene combinations is in the millions, but junctional diversity increases that dramatically. T-cell receptors reach an estimated total diversity of 10 to the 18th power, far exceeding that of antibodies. This extensive diversity ensures that both B cells and T cells can recognize an almost limitless range of antigens. T-cell receptors recognize peptide antigens bound to MHC molecules. This figure shows how T-cell receptors recognize antigens presented by major histocompatibility complex molecules. First, a pathogen protein inside the cell is broken down into smaller fragments through antigen processing. Next, one of these peptide fragments is loaded onto a major histocompatibility complex molecule inside the cell. The complex is then transported to the cell surface, where the peptide is displayed for surveillance. Finally, a T-cell receptor on a nearby T-cell binds to the major histocompatibility complex molecule and its bound peptide. This interaction is essential for T-cell activation, allowing the immune system to detect and respond to infected or abnormal cells. Two classes of MHC molecule present peptide antigens to two types of T-cell. This diagram compares the two classes of major histocompatibility complex molecules, class I and class II. Both types bind short peptide fragments, but they differ in structure, location, and the T cells they activate. Major histocompatibility complex class I molecules, shown on the left, are composed of a heavy chain and a beta-2 microglobulin. They present peptides to cytotoxic T cells. On the right, class II molecules consist of two similar-sized chains. They present peptides to helper T cells. Each class of molecule plays a critical role in antigen presentation and immune activation. This slide shows the structural differences between the CD4 and CD8 co-receptors, both of which assist T-cell receptors during antigen recognition. At the top, the CD4 molecule is made of a single chain with 4 extracellular domains, labeled D1 through D4. It is expressed on helper T cells and binds to major histocompatibility complex class II molecules. Below, the CD8 co-receptor is made of 2 chains, alpha and beta. It is expressed on cytotoxic T cells and binds to major histocompatibility complex class I molecules. Both CD4 and CD8 stabilize the interaction between the T-cell receptor and the major histocompatibility complex molecule and help initiate signal transduction for T-cell activation. This figure highlights how T cells interact with different target cells to produce specific immune responses. At the top, a cytotoxic CD8 T cell recognizes a virus-infected cell through its T cell receptor. After contact, it triggers the infected cell to undergo programmed cell death. In the middle row, a CD4 helper T cell contacts a macrophage that has engulfed a pathogen. The T cell releases cytokines that activate the macrophage, enhancing its ability to destroy the ingested microbes. At the bottom, a CD4 T cell interacts with a B cell that has captured and presented antigen. Cytokine signaling from the T cell prompts the B cell to differentiate into a plasma cell, which secretes large amounts of antibodies. These direct cell-to-cell interactions are essential for coordinating effective immune responses across multiple arms of the immune system. The two classes of MHC molecule have similar three-dimensional structures. This figure compares the three-dimensional structures of major histocompatibility complex class I and class II molecules. On the left, class I consists of a heavy alpha chain with 3 domains, alpha 1, 2, and 3, paired with a beta 2 microglobulin. On the right, class 2 includes two chains, alpha and beta, each contributing two domains. Despite differences in subunit composition, both types of major histocompatibility complex molecules form a peptide binding groove at the top, which holds the antigenic peptide. This structural similarity reflects their shared function, presenting antigens to T cells, though each class does so to a different type of T cell. MHC molecules are crucial for immune recognition and share a similar three-dimensional structure despite having different roles. On the left, we see a target cell presenting an antigen with a class I MHC molecule. This molecule includes three alpha domains and a beta 2 microglobulin. The antigen is displayed between the alpha 1 and alpha 2 domains. A CD8 T cell recognizes this complex. Its T cell receptor binds the antigen, while the CD8 co-receptor binds to the alpha 3 domain. On the right, an antigen-presenting cell uses a class 2 MHC molecule made up of both alpha and beta chains. The antigen is presented between the alpha 1 and beta 1 domains. A CD4 T cell engages this complex, with its receptor recognizing the antigen, and the CD4 co-receptor binding to the beta 2 domain. Though structurally similar, Class I and II MHC molecules guide different types of T cells to perform specialized immune functions. MHC molecules bind a variety of peptides. This slide shows the three-dimensional structures of MHC Class I and Class II molecules, which play essential roles in antigen presentation. At the top, the MHC Class I molecule is displayed. It consists of a single alpha chain forming the peptide binding groove, shown here in green. The antigenic peptide, shown in red, sits tightly within this groove. The closed ends of the groove restrict the length of peptides that can bind. Below, we see the MHC Class II molecule. Unlike Class I, this molecule has both an alpha and a beta chain, shown in yellow and green. Together, they form a more open peptide binding groove. This structural feature allows Class II molecules to accommodate longer peptides compared to Class I. Despite differences in structure and peptide binding capacity, both classes are critical for T cell recognition and immune defense. MHC Class I and MHC Class II molecules function in different intracellular compartments. This slide shows how MHC Class I and Class II molecules function in separate intracellular pathways. On the right, MHC Class I molecules are loaded with peptides in the endoplasmic reticulum. These peptides are typically derived from proteins degraded in the cytosol and transported into the urine. Once loaded, the MHC Class I complex moves through the Golgi apparatus and is presented on the cell surface for recognition by CD8 T cells. On the left, MHC Class II molecules follow a different route. They are loaded with peptides in specialized vesicles after the proteins are taken in through endocytosis. These peptides are processed in endosomes and lysosomes. The MHC Class II complex is then loaded with antigen and presented on the surface for CD4 T cell recognition. This compartmental separation ensures that the immune system can monitor both internal and external threats. Peptides generated in the cytosol are transported to the endoplasmic reticulum for binding to MHC Class I molecules. This slide compares the constitutive proteasome and the immunoproteasome, two protein complexes responsible for generating peptides in the cytosol. On the left is the constitutive proteasome, found in most cells under normal conditions. It contains catalytic subunits labeled beta 1, beta 2, and beta 5, and is capped at both ends by the 19S regulatory particles. These proteasomes degrade damaged or unneeded proteins into peptides. On the right is the immunoproteasome, which is formed in response to inflammatory signals, such as interferons. It replaces the standard catalytic subunits with LMP2, MECL1, and LMP7. The caps are also replaced with PA28 complexes, which enhance the speed and efficiency of peptide generation. The peptides produced by both proteasomes are transported to the endoplasmic reticulum, where they bind to MHC class I molecules for presentation to CD8 T cells. The immunoproteasome plays a key role in enhancing antigen presentation during immune responses. This slide illustrates how intracellular proteins are processed and prepared for presentation by MHC class I molecules. In the cytosol, proteins are broken down by a structure called the proteasome. This generates small peptide fragments, shown as red dots. These peptides must be transported into the endoplasmic reticulum, where MHC class I molecules are assembled. The transporter associated with antigen processing, or TAP, plays a key role here. TAP is a membrane protein that selectively shuttles peptide fragments from the cytosol into the lumen of the endoplasmic reticulum. Once inside, peptides can bind to newly synthesized MHC class I molecules, preparing them to be displayed on the cell surface for immune surveillance by CD8 T cells. MHC class I molecules bind peptides as part of a peptide loading complex. This slide shows the step-by-step process of how MHC class I molecules are loaded with peptides inside the endoplasmic reticulum. In the first panel, the MHC class I heavy chain is stabilized by a protein called calnexin. This stability is maintained until beta-2 microglobulin binds. In the second panel, once beta-2 microglobulin binds, calnexin is released. The resulting complex joins with several other molecules, calreticulin, tapicin, ERP57, and TAP, to form the peptide loading complex. In the third panel, peptides produced by the proteasome in the cytosol are transported into the endoplasmic reticulum by TAP. One of these peptides binds to the MHC class I molecule, completing its maturation. Finally, in the fourth panel, the loaded MHC class I molecule dissociates from the complex and is transported out of the endoplasmic reticulum to be presented on the cell surface, where it can be recognized by CD8 T cells. This carefully regulated process ensures that only properly folded and peptide-loaded MHC class I molecules are presented to the immune system. This slide explains how peptide length is adjusted during MHC class I loading. In the first panel, a peptide that is too long at the amino terminus binds to the MHC class I molecule inside the endoplasmic reticulum. In the second panel, an enzyme called ARAP, endoplasmic reticulum aminopeptidase, trims the extra amino acids from the N-terminus. This produces a peptide that is the optimal length of 8 to 10 amino acid residues. In the final panel, once the peptide fits properly, the stable MHC class I complex is transported to the cell surface. There, it presents the process antigen for recognition by CD8 T cells. This trimming step ensures that only correctly sized peptides are displayed to the immune system. Peptides presented by MHC class II molecules are generated in acidified intracellular vesicles. This slide illustrates how antigens are processed for presentation by MHC class II molecules. In the first panel, an antigen is taken up from the extracellular environment into an intracellular vesicle. Next, in early endosomes where the pH is neutral, proteases remain inactive, and the antigen stays intact. As the vesicle matures, it becomes more acidic. This acidification activates proteases, which then degrade the antigen into smaller peptide fragments. Finally, these vesicles containing peptide fragments fuse with vesicles carrying MHC class II molecules. This allows the peptides to bind to the MHC class II complex, which is then transported to the cell surface for presentation to CD4 T cells. This process is essential for detecting pathogens that originate outside the cell. Invariant chain prevents MHC class II molecules from binding peptides in the endoplasmic reticulum. This slide explains how the invariant chain regulates peptide loading in MHC class II molecules. In the first panel, MHC class II molecules are formed in the endoplasmic reticulum. The invariant chain binds to the peptide binding groove, blocking premature peptide binding. In the second panel, as the MHC class II complex enters vesicles, the invariant chain is partially degraded, leaving behind a small fragment called clip, which remains in the binding groove. In the third panel, clip continues to block other peptides from binding while the complex moves through the vesicle system. Finally, in the fourth panel, a molecule called HLA-DM facilitates the release of clip. This opens the binding groove, allowing antigenic peptides to bind to the MHC class II molecule, which is then ready for presentation on the cell surface. This stepwise process ensures that only properly processed peptides are presented to CD4 T cells. Cross-presentation enables extracellular antigens to be presented by MHC class I. This slide illustrates the process of cross-presentation, an important exception to the typical MHC class I pathway. Normally, MHC class I molecules present peptides derived from intracellular proteins. However, in cross-presentation, extracellular antigens, usually processed by antigen-presenting cells, are redirected to the MHC class I pathway. As shown here, antigens are taken up by phagocytosis and processed in the phagolysosome. Instead of being loaded onto MHC class II molecules, some of these peptides are transported to the endoplasmic reticulum. Inside the the peptides are loaded onto MHC class I molecules. These loaded complexes are then transported to the cell surface. Cross-presentation allows dendritic cells to activate CD8 T cells against pathogens that do not directly infect them, playing a crucial role in antiviral and antitumor immunity. MHC class I molecules are expressed by most cell types. MHC class II molecules are expressed by few cell types. This chart compares the expression of MHC class I and class II molecules across different cell types. MHC class I molecules, shown in the left column, are broadly expressed. They are present on nearly all nucleated cells, including immune cells like T cells, B cells, macrophages, dendritic cells, and neutrophils. Even non-immune cells, such as liver hepatocytes, kidney epithelium, and brain cells, express MHC class I, though at varying levels. Erythrocytes, which lack a nucleus, do not express either class. In contrast, MHC class II expression is much more restricted. It is mainly found on professional antigen-presenting cells, B cells, macrophages, and dendritic cells. Some T cells and brain cells can express MHC class II under specific conditions, but other cell types generally do not. This selective expression ensures that MHC class I presents intracellular antigens to CD8 T cells, while MHC class II presents extracellular antigens to CD4 T cells, maintaining immune specificity. The T cell receptor specifically recognizes both peptide and MHC molecule. This slide highlights the precision of T cell receptor recognition, which depends on both the peptide and the MHC molecule. In panel A, we see the three-dimensional structure of a T-cell receptor bound to an MHC class I molecule. The receptor includes alpha and beta chains that interact directly with both the peptide and the MHC. The peptide lies within a groove formed by the alpha 1 and alpha 2 domains of the MHC molecule. Panel B simplifies this interaction, showing that the T-cell receptor spans the peptide MHC complex, making contact with both the presented antigen and the MHC itself. Panel C offers a surface view of the complex, revealing how the T-cell receptor fits over the MHC molecule like a lock on a key. Panels D and E compare the structures of the peptide binding grooves of MHC class I and class II molecules. Despite their structural similarities, each class presents peptides to different types of T cells, CD8 or CD4, depending on the context of the immune response. This dual recognition of peptide and MHC ensures that T cells can accurately detect infected or abnormal cells while maintaining self-tolerance. This slide compares the antigen presentation pathways for MHC class I and MHC class II molecules. On the left side, MHC class II molecules present extracellular antigens. These antigens are taken into the cell through endocytosis and processed in acidified vesicles known as phagolysosomes. The resulting peptides bind to MHC class II molecules inside vesicles. These complexes are then transported to the cell surface to be recognized by CD4 T cells. On the right side, MHC class I molecules present intracellular antigens. These are typically viral or self-proteins that are degraded by the proteasome. The resulting peptides are transported into the endoplasmic reticulum by the TAP transporter. There, they bind to MHC class I molecules, which are then moved through the Golgi and presented at the cell surface for recognition by CD8 T cells. This summary highlights how the immune system monitors both internal and external environments through distinct but parallel MHC pathways. The diversity of MHC molecules in the human population is due to multi-gene families and genetic polymorphism. This slide illustrates the genetic diversity of MHC molecules, which arises from multi-gene families and extensive polymorphism. At the top, we see the 6 major human MHC class I isotypes. HLA, HLB, and HLA-C are highly polymorphic, meaning they vary greatly between individuals. HLA-E, HLA-F, and HLA-G are less variable and are considered oligomorphic or monomorphic. These isotypes help the immune system detect infected or abnormal cells by presenting intracellular peptides to CD8 T cells. Below, the human MHC class II isotypes are shown. HLA-DP, HLA-DQ, and HLA-DUR are polymorphic and present extracellular antigens to CD4 T cells. HLA-DM and HLA-DU are less variable and serve regulatory roles, such as facilitating peptide loading. This wide genetic variation allows populations to present a broad range of antigens, enhancing immune defense across diverse pathogens. But it also complicates organ transplantation due to tissue mismatch. This chart highlights the extraordinary genetic diversity of human MHC molecules, also known as human leukocyte antigens, or HLAs. On the top half, we see MHC class I loci, HLA, HLA-B, and HLA-C. These are highly polymorphic, with HLA-B having over 2,500 known allotypes. In contrast, other class I molecules like HLA-F and G have very few variants, reflecting more conserved roles in immune regulation. The lower half shows MHC class II diversity. Loci such as DPB1 and DQB1 are also highly polymorphic, with hundreds of different variants. DARB1 is especially diverse, with over 1,100 known allotypes. Other Class II loci like DRA and DOA have only a few variants and are more conserved. This extreme polymorphism allows for broad immune recognition across populations but presents challenges for tissue matching in transplantation. The HLA Class I and Class II genes occupy different regions of the HLA complex. This slide illustrates the genomic organization of the human leukocyte antigen, or HLA, complex on chromosome 6. The HLA complex is divided into three main regions: Class II, Class III, and Class I. On the left, the Class II region contains genes such as DP, DQ, DR, and the regulatory genes DO and DM. These genes encode MHC Class II molecules involved in presenting extracellular antigens to CD4 T cells. In the center is the Class III region, which does not contain classical MHC genes, but includes other immune-related genes involved in inflammation and complement activation. On the right, the Class I region includes the highly polymorphic genes HLA, B, and C, along with the less variable HLA, F, and G. These genes encode MHC Class I molecules, which present intracellular antigens to CD8 T cells. The physical separation of these regions reflects their distinct functions and evolutionary origins in immune defense. This slide highlights the genetic diversity found within the HLA-DR region of the MHC Class II complex. Every individual inherits the DRA gene, which is invariant. However, variability arises from the DRB gene family, especially DRB1, which is present in all individuals and is highly polymorphic. Some individuals also inherit additional DRB genes, such as DRB3, DRB4, or DRB5, depending on their specific DRB1 allele. These additional DRB genes are not present in everyone and contribute further to immune diversity. The different combinations of DRB genes between individuals enhance the range of peptides that can be presented to CD4 T cells, allowing for a more flexible and robust immune response. This variability also explains why matching for HLA-DR genes is critical in tissue transplantation. Other proteins involved in antigen processing and presentation are encoded in the HLA Class II region. This slide shows the extended gene map of the HLA Class II region, emphasizing that it encodes not only classical MHC Class II molecules, but also other critical proteins involved in antigen processing and presentation. On the left side, we see genes like tapicin, TAP1, TAP2, LMP2, and LMP7, which play essential roles in peptide processing and transport, particularly for MHC Class I antigen presentation. In the middle of the region are the classical HLA Class II genes, such as DP, DQ, and DU, including both alpha and beta chain genes. These encode the molecules responsible for presenting peptides to CD4 T cells. Additionally, non-classical Class II genes such as DMA, DMB, DOA, and DAB are present. These help regulate peptide loading by facilitating the removal of the clip fragment and stabilizing the MHC Class II molecule during antigen presentation. This rich and tightly packed genomic region reflects the complex coordination required for effective immune surveillance. MHC polymorphism affects the binding of peptide antigens and their presentation to T cells. This slide illustrates how polymorphism in MHC molecules influences antigen presentation. At the top, we see MHC Class I variability. The alpha 1 and alpha 2 domains form the peptide binding groove, where most of the genetic variation occurs. These red represent sites of amino acid differences across individuals. This polymorphism affects which peptides can be bound and presented to CD8 T cells. Below, MHC class II variability is shown. The peptide binding groove is formed by the alpha 1 and beta 1 domains. Just like in class I, the red dots mark regions of high polymorphism. These variations determine the peptide repertoire that can be presented to CD4 T cells. This high degree of variability allows the immune system to recognize a wide range of pathogens, but it also contributes to challenges in transplant compatibility and autoimmune susceptibility. This slide demonstrates how polymorphism in MHC molecules shapes their ability to bind and present specific peptides to T cells. At the top, we see examples of MHC class I molecules. The HLA-A2.01 molecule binds a peptide derived from HIV reverse transcriptase. This peptide contains preferred amino acids at specific anchor positions, particularly hydrophobic residues like leucine and valine, which enhance binding stability. Similarly, HLA-B27.05 presents a peptide from the influenza A nucleoprotein. It shows a distinct binding motif, favoring arginine at both the N-terminal and C-terminal ends of the peptide. Moving to MHC class II, we observe that HLA-2RB104 presents a self-peptide from the immunoglobulin kappa light chain. The peptide aligns within a groove formed by the MHC molecule, with variable amino acids at contact points that reflect the molecule's polymorphic nature. Another class II molecule, formed by HLA-DQA105.01 and DQB1301, binds a self-peptide from the transferrin receptor, further illustrating how peptide recognition is guided by specific amino acid preferences. These examples highlight that even small differences in MHC alleles can significantly influence which peptides are presented, ultimately shaping an individual's immune response. This slide illustrates the concept of MHC restriction, how T cells recognize antigens only when presented by a specific MHC molecule. In the first panel, a T cell successfully recognizes antigen X when it is presented by the HLA-A0201 molecule on an antigen-presenting cell. The T cell receptor binds both the antigen and the MHC molecule with high specificity, resulting in immune recognition. In the second panel, although the same antigen X is presented, it is displayed by a different MHC molecule, HLA-B5201. Despite the presence of the correct antigen, the T cell receptor does not recognize this MHC combination, and no immune response is triggered. In the third panel, the correct HLA-0201 molecule is present, but it is displaying a different antigen, Y instead of X. Again, the T cell receptor fails to engage properly, leading to no recognition. This example emphasizes that for a T cell to respond, it must recognize both the specific peptide and the exact MHC molecule presenting it. This dual specificity ensures precision in immune responses, but also contributes to individual variation in immunity. MHC diversity results from selection by infectious disease. This slide illustrates the concept of heterozygote advantage in MHC diversity, particularly in the context of peptide selection. Each circle represents the range of peptides that a specific MHC haplotype can present. Haplotypes 1, 2, 3, and 4 each bind a distinct but limited set of peptides. When individuals inherit two different MHC haplotypes, such as haplotypes 1 and 2, they gain a broader repertoire of peptide recognition, shown by the presence of two non-overlapping circles. This results in greater immune coverage against diverse pathogens. In contrast, if the two inherited haplotypes, like 3 and 4, have overlapping peptide binding specificities, the advantage is reduced. The overlap limits the diversity of antigens that can be effectively presented to T cells. This model highlights why individuals with a greater diversity of MHC alleles often have enhanced immune responses. It also explains how infectious disease pressure has contributed to maintaining MHC polymorphism in human populations. This slide shows how MHC diversity in a population can be shaped over time by infectious disease and changes in population size. In the first panel, a population emerges from a period of war, famine, and poor health. Repeated infectious disease outbreaks, such as smallpox and influenza, impose strong balancing selection, favoring individuals who are heterozygous at MHC loci. This ensures survival across a broader range of pathogens. In the second panel, the population stabilizes and decreases slightly in size, entering a peaceful era with good nutrition and effective healthcare. In this period, selection pressure is weak, and MHC diversity remains relatively stable. The third panel depicts a new epidemic, such as HIV/AIDS. Here, only individuals with a specific MHC haplotype, represented in blue, survive. This imposes strong directional selection, dramatically shifting the genetic makeup of the population. Finally, in the fourth panel, a catastrophic disease event reduces the population by 75%. Despite intense selection, all 4 MHC haplotypes are retained in the survivors. This illustrates how even under severe pressure, genetic diversity can persist, especially when survival requires different immune profiles. Together, these scenarios emphasize how both historical and emerging diseases have helped shape the evolution of MHC diversity in human populations. This slide illustrates two mechanisms that generate diversity within MHC genes: interallelic conversion and gene conversion. On the left, we see interallelic conversion. Here, segments of DNA are exchanged between two different alleles of the same gene, such as B5101 and B3501. The resulting recombinant allele, B5301, carries a new combination of exons that did not exist previously. This process increases allelic diversity within a population. On the right is gene conversion, where a segment of DNA from one gene, such as CW102, is copied into a different MHC gene like B1501. This non-reciprocal transfer results in a novel variant, such as B4601, with a patch of sequence from another gene. These processes contribute to the extensive polymorphism seen in MHC loci, enabling the immune system to recognize a broader array of pathogens and adapt to evolving infectious challenges. This slide presents pie charts illustrating the frequency of various alleles for different human leukocyte antigen, or HLA, genes across a given population. Each chart represents a different HLA locus. At the top, we see the class I molecules, HLA-A, HLA-B, and HLA-C. For HLA-A, the 204 and 24, 02 alleles are predominant. In HLA-B, the most common alleles include 39, 05, and 39, 09. HLA-C is dominated by the 702 allele, with several minor contributors like 1503 and 401. Below, the class II molecules are shown, including HLA-DRB1, HLA-DQA1, HLA-DQB1, and HLA-DPB1. For HLA-DRB1, 411 is the most frequent allele, while for HLDQA1 and DQB1, 301 and 302 dominate, respectively. A similar pattern appears for HLA-DPB1, with 302 being the most common. These charts highlight the allelic diversity within the HLA system and show how certain alleles become more prevalent due to evolutionary pressures such as disease resistance, population bottlenecks, and genetic drift. This diversity underpins the importance of HLA matching in organ transplantation and influences how individuals respond to infections and vaccines. This graph illustrates how MHC genotype influences the progression of HIV to AIDS over time. The y-axis shows the percentage of HIV-infected individuals who remain AIDS-free, while the x-axis measures the number of years since seroconversion, the point at which a person tests positive for HIV antibodies. Each curve represents a group of individuals with different MHC class I alleles. Those with certain alleles, shown in red, experience a slower progression to AIDS, maintaining higher AIDS-free survival rates for up to 2 decades. Individuals represented by the yellow curve show intermediate progression, while those in the blue group progress to AIDS more rapidly, with fewer than 20% remaining AIDS-free by year 10. This data underscores the critical role that MHC diversity plays in shaping immune outcomes. Specific HLA alleles can significantly influence the body's ability to control viral infections, providing a survival advantage in the face of chronic disease.